Okay. Good. So, it has been a while since I've done a revision stream, let alone actually revised. We have exams in just about 6 weeks, I want to say. And what's interesting about these exams is she has told us that it's going to be an AS exam, which means it's only going to be year 12 content that we need to revise. Obviously, I still want to revise for year 13 stuff so I don't forget it, but over the next couple of revision streams, we're going to go back and cover as many year 12 things as possible. So we're going to start today with the all reliable biological molecules topic. Then the week after, we'll cover cells, things like that, transport across membranes, and eventually we'll work our way towards what I would consider, probably the more difficult topic, things like inhalation, exhalation, the cardiac cycle, things like that. So we're working our way up from the relatively easy topics. We are kind of in a bit of a situation because one of our biology teachers is just off. She's been off for some time now, which is a bit worrying because she's the one who's going to be teaching us the genetic stuff and the ecology stuff, which are my least favourite topics. Which is a problem because I won't learn them very well if I'm having to teach them myself. So yeah, let's just see how we go with this. Just gonna go find some pens. I really ought to clean these pens. I have not used them in a long time. Just need a black pen. There we go. Okay, so we'll start off with carbohydrates. That camera is flipped, that's good. Oh my god. Been a while since I've done this. Carbohydrates. So. We should probably first discuss what a monomer is and what a polymer is. Now, there is a specific definition, but I can't recall it, so I'm going to do my best to recall it. I'm pretty sure a monomer is a small repeating unit, and a polymer is a large unit made of smaller ones, made of smaller subunits. So pretty much they're interchangeable in terms of how you define them. A monomer would be, an example of a monomer would be something like a monosaccharide. An example of a polymer would be a polysaccharide. So for example, glucose would be a monosaccharide. Maltose would be an example of a disaccharide, or So that's kind of what the definition is meant to be, but I will look at that further to see if I've missed a point. Because I know there's an exam question that they've asked like 3 or 4 times now, and that the exam question is literally, what is a monomer? And it's 1 mark, and for very quick reference, the papers are out of 80, I believe. So that's 240 potential marks. From what I recall, I think the essay question is about 30 marks, something like that. So in other words, is it out of 80 or 60? Actually, I don't seem to recall. Either way, you need about 50 to 60% for a B or an A. So even 1 mark is vital. And that could be 1 mark that I've dropped there just because I didn't nail the definition. So yeah, a monomer, small repeating subunit that combines to create a polymer. That's what I would describe it as, but that might not be 100%. So we'll work on that in a bit. So, carbohydrates. Let's start then. So carbohydrates, obviously the monomer version of it is going to be a monosaccharide and a polysaccharide. So that's how you would, go about naming them. The bond between the saccharides is going to be a glycosidic bond. It's not ester bonds, it's not covalent bonds, it's glycosidic bonds. You put this word in any question related to carbohydrates, you most likely have a mark, okay? There is, and I will do a whole stream on this eventually because it's a vital piece of knowledge. It's about having like a toolkit of these keywords at your disposal. So when you go into an exam and you see a question on carbohydrates, you can immediately pick out glycosidic, saccharide, polysaccharide, monomer, polymer. It's all about getting those keywords in because even a single keyword could be the difference between a mark, which is insane. It really shouldn't be, but that's how the mark schemes are written. So let's actually go into the general knowledge now about carbohydrates. We'll start off just with some generic, almost like sums. You got glucose plus glucose equals glucose plus galactose and glucose and I believe it's fructose. Actually been a while since I've done this, so I could be I think I get these mixed up a lot more than you would think. So glucose and glucose, I'm pretty sure is maltose. Glucose and galactose is lactose. And glucose and fructose is sucrose. Now, we could link this to the digestion topic very quickly if we wanted to. Because all of these have digestive enzymes, but they are broken down by. Maltose is going to be maltase. Lactose is going to be lactase. And sucrose is going to be sucrase. But that's just a bit of general knowledge there. Enzymes, again, coming back to this idea of keywords, you always talk about the ES complexes, you always talk about active sites, you always talk about binding, complementary structure, tertiary structure. Just waffle and you'll get marks. So, carbohydrates. We should talk about isomers very quickly. And this links to the idea of alpha and beta glucose. So, these molecules are meant to look the same. I know my artistic talent makes them look a little bit different, but if we assume the shape is the exact same, we have a hydroxyl there, a hydrogen there, hydroxyl there. So these molecules are examples of isomers because they have the same structure, the same... I don't recall the exact terminology of the word like isomer. I don't recall exactly what it is, but alpha and beta glucose are examples of it. They are made of the exact same molecules. They are made up of the exact same molecules, but arranged differently. So this is beta glucose. This is alpha glucose. And we can tell because beta glucose has the hydroxyl on the top right. In alpha, the hydroxyl is on the bottom right. So alpha and beta glucose are interesting because they are used to create some biological molecules, some interesting little things. I'm gonna leave out on the board there very quickly, but really there's not much there. We've just gone over some keywords for this topic. Monosaccharide, polysaccharide, glycosidic bonds, all of the different kind of compounds there, and the enzymes. So not much we've really learned there. But let's link it back now. Let's link it back to the three different types. So firstly, we have cellulose. We then have, and I think is it glycerol? No, glycerol. I always forget this one. What you call it. Glycogen? Shit, it's long branch chains, only in mammals, or like animals. Storage molecule. Long branch chains, only in animals. Shit, I don't recall it. I don't remember what it is. Is it glycogen? I'll put glycogen. And the third one is going to be cellulose starch. Now we can draw an invisible line here. Because it turns out, key piece of information here, cellulose is beta glucose, glycogen is, and starch is alpha glucose. So, cellulose and glycogen and starch. Okay, let's start off with cellulose because this is the one that I think a lot of people struggle with. And I can understand why, but I think it can be easy to remember. All you gotta remember with cellulose is this idea of neat, straight rows. It's this idea that they are long, unbranched chains that is the idea. You need to talk about the idea of cross links between these molecules, and these form strong microfibers. I think you spell microfibers like microfibrils, which is weird, but it's this idea that the hydrogen bonds form strong microfibers. Because they're cross-linked. Now, cellulose, the purpose of cellulose is in cell walls. It is technically a storage molecule. I don't know whether cellulose, insoluble cellulose, I don't know whether it's... I'm going to assume it's insoluble. It's insoluble and provides structure. So it provides structural support. That's why all of these chains are cross-linked, because it provides some strong support, okay? So that's cellulose. Used in cell walls for plants, it's insoluble and it provides structural support and rigidity. Next, we'll go on to glycogen. The idea you need to think for this one is long branched chains. So the molecule itself, I'm pretty sure, is quite a small molecule, but it has long branched chains. And remember, these are all made up of glucose. Glycogen is made up of alpha glucose. That's what all of these branches are. And the reason you would say that it's long and branched is because it increases surface area, which leads to a faster hydrolysis. And you can kind of blow up that a little bit by saying, basically, hydrolytic enzymes which sole purpose is to break down, this molecule. Hydrolytic enzymes will be able to act on the molecule faster because of its increased surface area. If it was a big circle, it would be harder, but because it's spread out everywhere with this large surface area, it means a faster rate of hydrolysis for the hydrolytic enzymes that need to break it down. So, cellulose, glycogen, and starch. Starch is a weird one because it's made out of two molecules, amylose and amylopectin. Now, I do not recall which one is which. I will be completely honest with you here. I don't recall. Starch is a storage molecule. It is insoluble. The idea you need to remember, though, with starch is when it is broken down, when starch is broken down, it becomes soluble. And as a result, starch will affect the water potential of cells when it is broken down into sucrose. That's just a very important thing to remember. When it's broken down, it will affect the water potential. I'm going to go quickly check the difference between amylose and amylopectin. I'm going to try not to use my computer much today, but so glycogen is branched every 10 monomers. Amylopectin is branched every 20. So actually, it's branched at a lot more frequent. But anyways, let me go back now. Amylopectin is branched, but it's a lot like smoother branches than this. Amylose is coil. It's a heliceal shape, or a coiled shape. And I should probably quickly reiterate that this one is found in plants, this one is found in plants. So, this makes it compact, good for storage. So that's about it. You have cellulose, glycogen, and starch. Glycogen, highly branched. I should probably specify the highly branched. That's about it for everything to do with this topic. That's about everything to do with carbohydrates, from what I can remember. I might have missed a very obvious thing, but I think to very quickly consolidate this knowledge, make sure we have not forgot anything. It's only been 20 minutes, so we might be able to get through half of the topic in an hour. I'm going to go on this really good website. this A-level biology specific website because they do some quizzes on every single topic here. So, there are 3 quizzes. Let's just rush through these, shall we? 17 questions. Okay. Carbohydrates are formed from which monomers? That's going to be monosaccharides. Glucose, galactose, and fructose are all examples of what common monomers? Monosaccharides, easy. What type of bond? It's going to be a glycosidic. What term describes a molecule formed by the condensation of 2, that's going to be disaccharides. The disaccharide maltose is going to be 2 glucose molecules. Condensation, remember, condensation uses a water molecule, it's for removal of a water molecule to combine them together. How is, and very quickly actually, a good example of this is going to be the structure of alpha and beta glucose. Let's just say for the purpose of our demonstration that it's like this. If we have them bonding together, that's going to be OH, that's going to be in fact, no. I won't show you guys this now. I'll wait till we get onto proteins and then I'll show, because it's a lot easier to explain then. How is the disaccharide sucrose formed? That's going to be condensation of glucose and fructose. Lactose is going to be glucose and galactose. Condensation of 2 glucose leads to maltose. Glucose and fructose will be sucrose. Glucose and galactose is lactose. Polysaccharides are polymers formed from a monosaccharide subunit. What type of bond is the glycosidic bond? It's not, is it a covalent bond? Wouldn't it just be a hydrogen bond? Pretty sure. What components are involved in a hydrolysis reaction to release a monosaccharide? Hydrolysis is the addition of a water molecule, so polysaccharide and water. Glycosidic bonds are formed by what type of reaction? A condensation. They're broken by hydrolysis. A monosaccharide would not be fructose. Which of these is not a type of polysaccharide? Maltose, amylopectin, glycogen, or cellulose. Aren't they all polysaccharides? Aren't they all polysaccharides? Because cellulose is all beta glucose, glycogen is all alpha. I'll put amylopectin. Let's have a look at how well we did. I'm going to put maybe 85%, 80. Yep, 82%. Which ones did we get wrong? What type of bond is formed? Oh, it is a covalent bond. Interesting. I didn't know that. What is not a monosaccharide? Oh, yeah. Okay, I misclicked for that one. Maltose is not a polysaccharide. Because it's a disaccharide. Of course it is. Oof. That's a bit rough. 80% though, that's fine, I'll take that. Very quickly we'll do this one. How many questions? 25, oh come on. What is the term used to, okay, a molecule with a different orientation is an isomer. The structure of alpha glucose is the OH group is on C5 is down, because isn't it clockwise? So C1 to... So C1 is down, I'm going to say. Which isomer joins by a condensation reaction to form cellulose? Beta. Alpha. Which of the following are formed by condensation of alpha glucose? It's starch, glycogen. Oh, and fructose? Fructose is just independent though, isn't it? Like that, it's just doing its thing. Which of the following is... That's going to be 1. Which type of glycosidic bonds are seen in glycogen? No idea. We don't cover this, really. I'll just put 1 to 6. Which of the following is formed from an alternately rotated beta-glucose? That's cellulose. I forgot to mention that. Cellulose alternately rotate, so it's like flipped each other molecule. Which of the following is formed from entirely straight unbranched chains? That's cellulose. Strongest structure, I'll have to go with cellulose again there. Highly branched is going to be glycogen. No idea. Which statement is not correct with amylose? Amylose is probably not soluble. Yeah, it has to be insoluble because it doesn't affect the osmotic potential. Which types of following bonds are seen in the amylopectin molecule that makes up starch? No idea. Literally no idea. We've never done this. Which structural feature of glycogen allows for the fast breakdown? Highly branched. What type of bonds hold the beta-glucose chains together? That's hydrogen. Which of the true is not following? They are not highly branched. What type of reaction breaks down glycogen for respiration? That will be hydrolysis, obviously. Cellulose. What molecule is branched? Amylopectin. What is the best description of glycogen? Short, highly branched chains. Best description of cellulose, long, straight, unbranched. Amylopectin is long, hold on. Long branched chains. Which molecule forms fibers? Cellulose. I'm going to put maybe 70% there. There were a few I didn't know. 92%. Okay, cool. The only ones I got wrong were the ones talking about specific bond lengths, which we've never covered. Final one is reducing sugars test. This one should be easy. It's the Benedict's test. Benedict's solution is, I think it's copper sulfate, maybe. Why is sucrose known as a non-reducing sugar? So I forget this. There are sugars like raffinose, which are non-reducing. Sucrose and raffinose. So sucrose is a non-reducing because it does not reduce copper sulfate. In order to do that test, you would first need to add an acid, then neutralize it, and then perform the Benedict's test as normal. Because you're adding the acid to break apart the glycosidic bonds. Positive result for the Benedict's test is it turns brick red when heated. Or it turns orange or green, really. What do you need to do to non-reducing sugars? Hydrolyze into its monosaccharides. Testing for starches with iodine. And it's potassium iodide, technically, and it goes blue and black. What colour is seen with a Benedict's test when there is a trace amount? Green. When there is none, it's going to remain that blue, that nice copper colour. What colour is seen with the Benedict's test when there is a moderate amount? I'd go with orange there. When there's large amount, it's brick red. Brick red again. What technique could be used to separate a mixture of monosaccharides to identify their components? Separate a mixture? Filtration, I'd assume. Reduction is the chemical reaction involving... Reduction is the gain of electrons. Reducing sugars donate their electrons to Benedict's reagent. I think it's copper 2 oxide. To test for reducing sugars, a food sample is ground up in water, mixed with Benedict's reagent, and then heated gently. Potassium iodide is used to test for starch. The test for starch is carried out, I don't think it matters. I'd say room temperature. Food must be ground up in water. For an emulsion test, it should be ground up in ethanol, I believe. Copper 1 oxide. Oh no, so I got that wrong. It's copper 1 oxide, not 2. Copper 1 oxide formed when Benedict's reagent is incubated with a reducing sugar. It's a insoluble red precipitate. Gonna say 70% here, maybe. Oh yeah, 90 does a good job as well. Oh, you would use chromatography. Okay, cool. Well, there you go. I'm happy with that. I think we've covered that whole topic very nicely there. It's getting a bit dark. I'm wondering, do I want to continue talking about proteins for an hour? Or do we head off now? I take the rest of the day just to chill, and then tomorrow we do... Because the thing is, with these revision streams, I honestly prefer them being short, because we can bam through a lot more topics. Better to have half an hour of hyper-focused revision on one topic than 2 hours where we go through like 5 topics and get lost along the way. So you know what I might do? I might wrap up the stream now. Might come back on later tonight to do part 2 where we cover proteins. And then, I mean, already, look, we got the biological molecule topic. Carbohydrates. Lipids. Which would only take like half an hour, lipids. Proteins. Enzymes. DNA. RNA, which also links to translation. And that'll be its whole other stream. Translation and transcription. Water. ATP, which I've already done a stream on, and ions. So, looking at it this way, we have just spent half an hour doing focus revision on carbohydrates. We can cross that one off. We have done a stream on ATP already, so we only have these left to do. I could easily do a stream tonight on lipids. We'll do proteins tomorrow. And with that, we can also do enzymes as well. DNA and RNA will be a separate stream entirely. And water and ions we could easily do on Tuesday. Or Wednesday. So we'll get this whole topic done. We'll get this whole topic done by the end of the week. And then that's us done with what I would argue is the hardest topic. Biological molecules, everyone struggles with. Even I did struggle with it, but now I find it a breeze. The idea with this topic you really need to nail in your head is it is unforgiving as hell, but once you get it, you cannot unforget it. And the idea is every single thing links back to this topic, okay? Look at flipping respiration and this idea of, talking about reduction, oxidation. You need to know your basic things about ions there. Water, when you look at photosynthesis, or when you look at the columns in transpiration, even that links very well. DNA and RNA, there's a whole topic on those, proteins and enzymes. They always ask about enzyme control reactions in the exam. Carbohydrates and lipids don't get much love in the exam. That's why I've only spent half an hour today doing focus revision. They don't get much love, but there was that question on raffinose, which everyone struggled with. Very quick tip, actually. Let me give a quick tip. Remember, a condensation reaction involves the addition of H2O. The removal. So if we had a molecule which was, I don't know, I'm gonna make these up, so it could be scientifically wrong. 484, and let's say it was a quad saccharide. I don't know if that's a thing. Well, it will be, but you know what I mean. So you would multiply all of this by 4. Okay, so 16, 32, 16. But the last thing you need to do is, because it's a 4 times, that's going to be H2O. So that'll be minus H8 minus O4. So what we actually get left with is C16 H24 O12. And that is the molecule that we'd be left with. There are so many questions that you could get asked where it'll ask you to do that basic equation that people will get wrong, because all they'll do is multiply it by 4 and forget to take away the H2O. Always take it away.